One is galactose. Galactose is a monosaccharide, which is a simple form of carbohydrate. It is hexose sugar with the molecular formula C6H12O6. Then, galactose is naturally found in milk and dairy products as a part of lactose. Galactose plays an important role in energy metabolism and biological process. How galactose is made? Galactose is mainly produced in the body from lactose, which is a sugar found in milk and dairy products. When we consume milk, the enzyme lactase breaks lactose into two simple sugars, glucose and galactose. The galactose produced is then absorbed into the bloodstream and transported to the liver. In the liver, galactose is converted into glucose or used to produce energy and other important biomolecules. Galactose can also be synthesized naturally in the body through biochemical pathways where it is used to form glycoproteins and glycolipids, which are important for cell structure and function. Is galactose safe to consume daily? Galactose is generally safe to consume daily because it is a natural sugar found in foods. In the body, galactose is normally converted into glucose in the liver and used as a source of energy or for important biological function. However, galactose is usually consumed as part of lactose, not on its own. When taken in normal dietary amounts, it does not cause harm to healthy individuals. An exception is people with galactosemia, a rare genetic disorder. They cannot properly metabolize galactose, so it must be avoided in their diet. Overall, for healthy people, galactose from normal food sources is safe to consume daily in moderate amounts. Structure of galactose. First is structure of 2D visual projection. A visual projection is a two-dimensional drawing used to represent the three-dimensional structure of a molecule. It is commonly used for molecules that contain chiral carbon atoms. The structure of a visual projection is drawn in the shape of a cross. At the center of the cross is a carbon atom, usually a chiral carbon, bonded to 4 different groups. Second is the structure of 2D Harvard projection. This structure shows galactose in the Harvard projection, which is a 2D presentation of its cyclic form. In the Harvard projection, galactose is drawn as a six-member ring, also known as Pyranus ring, made up of 5 carbon atoms and 1 oxygen atom. The ring is drawn almost flat to help us clearly see the position of each substituent group. Lastly, the structure shows galactose in a 3D ball and stick model, which represent the actual three-dimensional arrangement of atoms in the molecule. In this model, each ball represents an atom such as carbon, oxygen, or hydrogen, while the stick represents the bonds connecting these atoms. Galactose forms a ring structure, specifically a six-member ring consisting of 5 carbon atoms and 1 oxygen atom. The 3D ball and stick model clearly shows that the molecule is not flat. The atoms are arranged at different angles, allowing us to see the true spatial orientation of the OH groups around the ring. Type of general properties is monosaccharide, which has chemical formula C6H12O6, which is glucose, or specifically an primer at C4. It has molecular weight 180.16 gram per mole, and it can be found in milk, some fruits, and also vegetables. Besides that, the chemical properties can reduce in sugar because it contains a free aldehyde group in the open chain. It can also undergo oxidation to form galactonic acid, and it also can participate in glycosidic bond formation. So, appearance of physical properties is white and crystalline solid. It can be soluble in water and also slightly soluble in alcohol. It tastes sweet but less sweet than glucose. The melting point is around 167 until 168 degrees Celsius. Besides that, the biological properties is a component of lactose. It also can be converted in a liver to glucose for energy. It is very important in glycoprotein and glycolipid synthesis. It can be defects in galactose metabolism cause galactosemia, which can lead to liver damage, cataracts, and also developmental issues. The diagram illustrates the Delayer pathway, which is the primary pathway for carbohydrate synthesis, and interconversion in which galactose is converted into glucose. In this process, galactose is first phosphorylated by galactokinase using ATP to form galactose 1-phosphate. This compound then reacts with UDP glucose via galactose 1-phosphate uridyl-transferase to produce UDP galactose and glucose 1-phosphate, which can enter glycolysis or glycogen synthesis. UDP galactose is subsequently converted back to UDP glucose by UDP glucose 4 apomerase, a reaction involving NAD+ or NADH, and inversion of the hydroxyl group at carbon 4. Overall, this pathway enables dietary galactose to be efficiently utilized as glucose in normal carbohydrate metabolism.
The use of galactose in food is galactose can be found in fermented products such as yogurt and kefir. It can also be found in very small quantities in artichokes, mushrooms, papayas, and tomatoes. In agriculture, galactose is a component of galactomannase, which is part of plant cell wall polysaccharides that contributes to plant cell structures and growth. In biotechnology, galactose is used to generate galactose-containing conjugates to improve drug delivery and absorption. For example, it is attempted for dopamine supplementation in Parkinson's patients. In pharmaceutical, galactose aids in treating galactosemia, a rare genetic disorder where the body cannot properly metabolize galactose. It is also have been used in treatment and diagnosis of hepatitis C, hepatitis B cancer, Wilson's disease, diabetic macular edema, and focal segmental glomerulosclerosis. In nanotechnology, galactose-containing nanoparticles have been developed that are selectively taken up by liver-related cells, showing potential for targeted drug delivery. For biofuels, advanced biofuels that were synthesized from galactose could replace gasoline, diesels, and even jet fuels. Advanced biofuels also has the potential to be carbon neutral. Furthermore, for green chemistry, plant polysaccharides like hemicellulose that includes galactose units are renewable bio-based materials in green chemistry to replace petrochemical polymers. Lastly, for sustainable materials, plant-derived polysaccharides that include galactose containing like hemicellulose are used as biodegradable polymers. Firstly, based on nutritional value, galactose are available as a dietary supplement. Next, some risk of galactose is that galactosemia can cause severe complications, including liver damage, intellectual disability, cataracts, and even death. Excessive intake of galactose may cause some adverse effect when consumed in large amounts over a prolonged period. Lastly, from the health benefits, galactose supplementation under medical supervision can improve glycosylation and clinical markers in specific metabolic disorders. It may also enhance oxidative metabolism in muscle cells. From Global Health Challenge, excessive intake of galactose, particularly from supplements, can lead to gastrointestinal discomfort, including bloating, diarrhea, and abdominal pain. High level of galactose have been associated with oxidative stress and inflammation, which can potentially damage cells and tissues, contributing to a variety of chronic health conditions such as cardiovascular disease and diabetes. Thank you.
In vivo mouse experiment, mice were fed either a high galactose diet or a normal diet. Tumor models including MC38 colon cancer and B16F10 melanoma cells were established to examine the effect of galactose on tumor growth and immune responses. Tumor infiltrating CD8 positive T cells were then analyzed by flow cytometry to assess exhaustion, proliferation, and cytokine production. Secondly, for molecular and cellular studies, gene and protein analyses such as RNA sequence and immunoblotting were used to measure IGF-BP1 expression and MTORC1 signaling activity in the liver cells. Genetic mouse models with IGF-1 receptor deficient T cells were used to mimic the effect of high galactose on T cell exhaustion. Finally, human cancer data were analyzed by correlating plasma IGF-BP1 levels with T cells exhaustion in human tissue. In vivo mouse experiment, mice were fed either a high galactose diet or a normal diet. Tumor models including MC38 colon cancer and B16F10 melanoma cells were established to examine the effect of galactose on tumor growth and immune responses. I will move on to the main findings of this study, which can be summarized into three key areas. First, regarding dietary galactose and anti-tumor immunity, mice that were fed a high galactose diet showed significantly stronger anti-tumor CD8 positive T cells responses compared to the control group. These mice also exhibit reduced tumor growth. Importantly, high dietary galactose prevented CD8 positive T cells exhaustion, allowing these immune cells to remain functionally active in targeting tumor cells. The second key findings involved hepatocytes reprogramming. Circulating galactose was found to reprogram hepatocytes metabolism by specifically inhibiting MTORC1 signaling in the liver. This metabolic change led to an increased production of IGF-BP1 or insulin-like growth factor binding protein 1 by hepatocytes. Finally, the group examined the role IGF-BP1 in the T cell function. IGF-BP1 acted to restrain IGF-1 signaling, which is known to promote T cell exhaustion. Supporting this mechanism, mice with T cells that lack of IGF-1 receptor showed similar improvement in the T cell function to those observed in the mice fed a high galactose diet. These findings indicates the blocked IGF signaling is a central mechanism behind the enhanced anti-tumor response. Importantly, these findings were also supported by human data. In cancer patients, high plasma levels in IGF-BP1 were associated with reduced T cells exhaustion and enhanced T cells activity within tumor tissues. Overall, these results suggested that dietary galactose enhances anti-tumor immunity through liver-mediated metabolic reprogramming and regulation of IGF-1 signaling. Next, I will briefly describe the experimental method used in this part of the study. First, adaptive laboratory evolution was carried out to obtain strains that were able to efficiently utilize galactose. Through repeated culturing under selective condition, strains with improved galactose metabolism were developed. To evaluate their performance, galactose consumption and cell growth were monitored. These measurements were carried out using spectrophotometry to assess cell density and high-performance liquid chromatography, or known as HPLC, to quantify galactose level. Following this, whole genome sequencing was performed on the evolved strains to identify genetic mutations that may be responsible for the improved galactose utilization. In addition, gene expression levels were analyzed using quantitative RT-PCR to determine changes in the transcription of key metabolic genes. Finally, enzyme activity assays were conducted to measure protease and beta-galactosidase activities using standardized assay kits, allowing assessments of functional changes at the enzymatic level. Together, these methods provided a comprehensive understanding of the genetic, transcriptional, and enzymatic adaptations involved in galactose utilization. First, the evolved strain showed noticeably faster growth when grown on a galactose compared to the wild-type strains. This indicates that the adaptive laboratory evolution process successfully enhanced the ability to grow using galactose as a cover source. In addition to faster growth, galactose utilization efficiency was significantly improved in the evolved strains, showing that these strains were able to consume galactose more effectively. Whole genome sequencing revealed key mutation in regulatory genes associated with carbon metabolism. These mutations are likely responsible for the improved metabolic performance observed in the evolved strain. Gene expression analysis further supported these findings, as genes evolved in galactose metabolism were expressed at higher levels in the evolved strains compared to the wild type. Finally, enzyme assays showed increased enzyme production in the engineered strains, indicating enhanced metabolic and functional capacity. Overall, these results demonstrate that adaptive laboratory evolution successfully improved galactose metabolism through genetic, transcriptional, and enzymatic changes.